Good afternoon and good morning, good evening to all of you, wherever you are. It's a pleasure to be chairing this fantastic panel discussion. We will be talking about cellular models for M2M2M. And we have four great panelists, and I will let them introduce themselves and tell us a little bit about their expertise. Helena, could you start?
Sure. Well, hello, everyone. My name is Helena Kilpinen. I'm an assistant professor at the University of Helsinki, based at the Institute for Molecular Medicine Finland, or FIM. I have worked for a long time on human-induced pluripotent stem cells. I was part of the big human-induced pluripotent stem cell initiative in the UK, where a large panel of iPSCs were generated and characterized. And my group has since worked on iPSCs, or used iPSCs to model neurodevelopment as well as neurodevelopmental disorders. And specifically, we do things like pooled culture of these cells to try to scale up experiments. And specifically, we're trying to understand and control for the various differences in these approaches, including differentiation efficiency.
Thanks very much. Patricia?
Hello, everyone. I'm Patricia Severino. I work at Albert Einstein Hospital Research Institute in São Paulo, Brazil. We are mainly interested in gene expression regulation at different levels. We address cancer biology, including biomarker discovery in DNA, RNA, and microRNAs, and we work with different cell models for the understanding of mechanisms in these two areas.
Thanks very much. I'll ask David to introduce himself next.
Hello, everyone. I'm Dave Van Valen. I'm an assistant professor at Caltech in the Division of Biology and Bioengineering. And I'd say what's relevant for this discussion is my lab spends a lot of time developing new deep learning methods for analyzing images of cells, in particular, both cells and tissues, and then also cells in live cell imaging data.
Thank you. And last but not least, Melina.
Yeah. Great, thank you so much. This is wonderful to be part of this panel. Thanks for the invite. So Melina Claussnitzer, I'm a faculty member at Harvard Medical School, based at the Center for Genomic Medicine and the Endocrine Division at MGH, and my lab is located at the Broad Institute. So my team is historically deeply invested in developing and applying experimental and computational strategies for variant to function studies in the context of metabolic disease, and really establish causal links between genetic variation, enhancers of actions, target genes of actions, the cell context these variants act in, and morphological and cellular consequences to basically develop actionable therapeutic hypotheses. So we primarily and historically work with adipocytes as model systems and have both develop or spend a lot of time in developing these model systems and optimizing them to faithfully recapitulate physiology that is relevant to metabolic disease. And we also developed several different image-based technologies to read out phenotypes of these cells.
Thanks very much. I should have introduced myself. Gosia, Trynka. I run a research group at the Sanger Institute in Cambridge, UK, and I'm also Director of Experimental Sciences of Open Targets. For all the participants, we encourage you to use Slido and ask questions to the panel. And perhaps to give you some food for thoughts, I'll start off with a question that I'd like to ask each one of you in the panel. And that is, within your domain, if you could tell us, how do you think about what defines a robust cellular model? And perhaps I'll start with Melina.
Sure, yeah, happy to. So in our case, we mostly realized for adipocytes that we were required to develop, or in order to achieve, to have a high confidence model system for variant to function studies, that it really requires to build a biobank of primary adipose-derived mesenchymal stem cells from different adipose depots. We mostly work with visceral and subcutaneous adipose tissue, which we retrieved from many hundreds of fully genotyped donors. We built this biobank in collaboration with Hans Hauner at the Technical University of Munich. And these differentiating adipose-derived mesenchymal stem cells allow to study cell state transitions, differentiation dynamics of adipocyte functions in the context of natural genetic variations. So these cells can be, exposed to stimulatory conditions to study diverse cell contexts and are editable for CRISPR studies. So from our experience, which really in a nutshell is that it takes time and efforts to develop a model system for variant to function studies that faithfully recapitulate the physiology of, in our case, metabolic disease. And to optimize our model system, what we have done so far and would advocate for is to first identify cell states faithfully at the tissue level and basically identify these cell states and molecular signatures in vivo through deep molecular profiling and develop these gold standards and reference data sets from the tissue level cell state, basically. And then use these molecular profiling data combined with enrichment type of analysis to optimize differentiation cultures in really iterative processes. So we have spent quite a lot of years to do that. And the outcome is, in recent work with Evan Rosen, as an example, Marco Emond is a postdoc who has been working on this, is that we can say from single cell RNA sequencing based profiling of tissue, we can recapitulate 7 out of 8 adipocyte cell states. The one that we cannot recapitulate is cell-cell interactions, but the rest through these iterative processes we can confidently recapitulate, which I think is the outcome of a long journey and is giving us confidence that it was worth it.
Great, thank you. I'll maybe move on to Helena and hear from Helena about iPS cells and modeling neuronal tissue, neuronal cells.
Sure. I think lots of the things Melina raised in a way apply also to the iPS world. iPS cells have been around now for quite a while. And they're used a lot, but it wasn't until fairly recently when the kind of large-scale characterization of them started. And thanks to that, we now have multiple big biobanks containing very well-characterized iPS cells. So that's great. I think in terms of characterizing these cells for a long while, the focus was on kind of confirming pluripotency, making sure they are pluripotent, and also karyotypically normal. But I think what we're learning and seeing is that it is very important to sequence your cell line to know what genetic variants are there, either in the germline or somatically acquired during the various times these cells spent either in reprogramming or in culture otherwise. So kind of knowing exactly the genetic background of each line is becoming more and more important, and it's not something that's systematically yet done in the field, I would say, partially because it's still expensive. The other thing is that differentiation of iPS cells is a noisy process, as anyone will know who's attempted that, regardless of the cell type you're interested in. And similarly, in that realm, we're only beginning to understand where or what causes the variability in those processes, so variable differentiation outcomes, for instance. Scale is an issue. So a lot of groups, including ours, have turned to these pooled systems where you co-culture and co-differentiate cells, and that brings yet another layer of complexity to these experiments because cells obviously interact in the dish. So I think overall, in terms of reliability, there has been some attempts in the stem cell field and more broadly to kind of start establishing community standards of how one might or how one should characterize each and every IPS line that is being used. But I think those haven't yet become kind of ubiquitously adopted, which means there's quite a lot of variability in the results that are coming through from different IPS-based studies.
Patricia, from your perspective, could you tell us how do you think about defining a robust cellular model?
Maybe to keep in line with what they have been talking about, we work a lot with macrophages and dendritic cells from lymph nodes in association with inflammation in cancer and other inflammatory diseases. And we work with different donors. So maybe I would add that we always go for a panel of markers that we have established that should be expressed at regular levels so we can have comparable results between different donors. We don't work with established cell lines. They are always from different donors. And since we do want to understand biological differences also between the donors, when we are looking for a model for a specific mechanism we're trying to understand, for example, differentiation, macrophages, and dendritic cells, we look for the specific panel of expression levels of markers. For example, cell receptors, internal or membrane receptors, for example, which are very important for the mechanisms that we're studying. So I believe that understanding which mechanism is of interest and which markers are of interest for studying those specific pathways or cellular processes is very important. And considering what also has been said, that cells are going to respond differently on the dish from what you were expecting, getting the cells originally from the donors, this subset of characteristics are very important. We do the same when we are expanding cells in bioreactors. We also have specific profiles that we identify prior to having the cells used for the expansion, and then we check for these markers later on to see if they kept those specific markers too. So I believe those are important issues that we should consider when we're looking for robust models, and it doesn't really matter which cell line we are or which cell origin we are working with.
Great, thank you. And David, from your perspective, how do you think about robust cellular models?
One, I would just echo everything that everybody in the panel just said. And I'd say, like, for our purposes, really what we look for are cellular models that have robust and quantifiable, like, visual phenotypes, so that you can image them and then identify those phenotypes, fairly reliably using modern machine learning algorithms. I'd say, like, that's for us, like, that's the aspect of, like, robustness that we're the most interested in. Although everything that everyone else has said almost certainly matters.
Thank you. So I do hear reproducibility, variability are some of the challenges, and then using the right reference panels to which one is comparing what's being modeled in a dish. And along those lines, there's a question that's, I guess, addressed to those of you working with IPS cells, and perhaps a bit more specifically to Melina. So I'll address it to you, Melina, but Helena, please do feel free to pick up afterwards. The question is, how faithful are IPSs in modeling their end cell equivalent in tissues compared to primary cells? And also, what is scalable and reliable? So in other words, for instance, adipocytes derived from stromal vascular fraction versus primary adipocytes versus iPS-derived iPSCs.
Yeah, that's a great question. So just flagging, we are only working with primary cells. We actually started early in my personal career before starting the lab. We have done iPSC-derived adipocyte differentiation protocols, and we were not very successful. The variability was really high, and we were not able to recapitulate more mature cellular states in these cells. In terms of the stroma vascular fraction and floating mature adipocyte in tissues, that's a very good question. We do not work with the stroma vascular fraction itself, but we basically proliferate the cells and enrich for progenitors that recapitulate the adipocyte progenitor state rather than the crude heterogeneous stroma vascular fraction, which we find to be a very important point. What I had mentioned before, this kind of enrichment analysis and benchmarking against the single cell RNA sequencing data from adipose tissue, which basically are molecular profiling data sets from the mature adipocytes, the floating adipocytes as they are present in adipose tissue. We are recapitulating all but one cell state, and the one cell state we cannot recapitulate is the one that is characterized by basically cell-cell interaction. So that we are not able to pick up in our in vitro system, and for that, I think studying floating adipocytes, which we also have done quite a bit early in our efforts, is critical and really a beautiful addition to the in vitro differentiation kind of model system.
Helena, do you want to maybe comment on this a little bit?
Yeah, perhaps a quick comment. Just in the neuron world, this is certainly a concern. That how mature are the neurons we produce in the dish? And I think there has been a number of examples where it has actually been concluded that what we're producing in the dish is more closely related to fetal tissue than necessarily adult tissue. So this is a concern. Again, could be sort of and should be addressed with different types of benchmarking approaches. In the field of from my perspective, studying neurodevelopment and developmental disorders, this may be less of an issue. We probably want to focus on more kind of early and less mature neurons. But of course, if you're modeling something like neurodegeneration, this is not true. There has been some or good examples of kind of improving the maturity of iPS-derived neurons by engrafting them into, say, mouse brain. This has helped. But of course, this adds another layer of complexity to any experiment and modeling study.
Since I have you here, the other question is if you're aware of any systematic comparisons of the transcriptome to of differentiated iPS cells to primary cells. I'll ask this question, Helena, to you, but if anyone on the panel is aware of such resources, please also take place to speak up.
Sorry, resources for that or
systematic comparison,
systematic characterization.
Mm-hmm.
I'm not aware of any systematic efforts. Of course, anyone who will use single cell approaches to profile their iPS-derived cells, and this is most of us these days, because heterogeneity is high, then of course you would leverage external resources, for instance, from the Human Cell Atlas or equivalent single cell resources to annotate your cells that you're seeing. So I suppose this is what you would call that kind of characterization. But more generally, I don't think it has been done in any kind of more organized way. It's kind of each study for themselves.
Mm-hmm.
Maybe just adding here, or not really adding, actually just echoing, I think from my perspective, also this is really lacking in the community, the kind of benchmarking of primary differentiation system and iPSC-derived differentiation system. And I think we as a community, it would be really valuable to do that because it could, serve to improve iPSC differentiation protocols, just develop overall better models for the entire community for different diseases. Making that happen would certainly be really valuable from, primary cells and iPSC derived cells from the same donors and same individuals, basically. So there's an underlying identical genetic background.
And perhaps if I can...
Sorry, if I may just follow up on what Melina said. I think I totally agree. I think this kind of benchmarking would also address the issue of having just tons and tons of different types of differentiation protocols aiming to derive the same cell type in the end. And if I don't think we'll eliminate this. People like their protocols and like to tweak their own protocols. But if at least we could, as a community, agree that in the end, for this type of tissue or cell types, this is what it should roughly look like, I think that would already be hugely useful.
Mm-hmm. Yeah, so I hear like a workflow of benchmarking steps. So one, if perhaps not even agreeing on the protocols, one could say what a certain cellular model is capturing in respect to a given reference. And I guess we kind of by default very often rely on the gene expression, for example, for these comparisons or surface markers. And I want to direct this question maybe to David and also to Patricia. David, how much more value is there in adding in other sources of information for cell phenotyping from your experience, in addition to transcriptomics?
I think it depends on the system, but there certainly is some value, and I think there are some things that you can measure with imaging that are quite hard to measure with some of the more omic approaches like transcriptomics. In our lab, we use the live cell imaging technologies to measure the dynamics of the cell signaling state, and that's something that because omics is sort of like a fixed endpoint measurement, you're just not going to be able to capture. So I would say the additional piece that can be hard to capture with the omics is also the cellular phenotypes of tissues as well. Although there are a increasing number of spatial omics technologies that are able to do this, I'd say the traditional way of doing it has been more with imaging. So I think it depends. I personally don't view them as being in conflict, but rather I view the two approaches being complementary. And we're spending increasing amounts of time thinking about how can you merge the information that you learn from imaging with the information that you've learned from some of these other assays like single cell RNA sequencing.
Mm-hmm. And Patricia, would you like to add anything here?
Yeah, we're just starting with single cell transcriptomic and single cell imaging. So we do not have a lot of expertise on this, but what we have been able to see is that even though we don't have such granular level information with the imaging that we usually do on tissues using also confocal microscopy and other systems like that for protein level assessment, we have also been able to validate results that we have from transcriptomics and to characterize the cell types that we are using in our models. And what I would like to point out is that there are subsets of characteristics and phenotypes that cells, I do not work with IPS so much undifferentiated cells, but working with more differentiated cells, some phenotypes are kept when we are working on the dish, and sometimes this can be very surprising. So what I believe we don't have is this subset of phenotypes that you can actually keep when you are working with the dish. We are many times surprised with cells, immune cells, which are circulating in different patients, keeping the same characteristics when we are working with them on the dish. And with the dish, and at times this varies. At other times they're very stable. And I think that knowing what you can really replicate on the dish is really important. And we have very little information about that. So what are the limits of which kind of phenotypes are going to be kept when you're using those cells for specific mechanisms? And this, I think that in line with what the girls were saying, could contribute to the harmonization of protocols, like having a better idea of the limitations of the models and how you can grow from those.
Great. Let me turn to our chat. There's a question from Tuli Lapalainen asking, how important do you think it is to study cellular models from multiple individuals if one wants to use them, for example, for CRISPR or drug perturbations? For which questions would one canonical model per cell type or cell line be enough? Anyone wants to tackle this question?
Happy to take a first step here. It's actually a really complex and very good question. So first off, in terms of drug perturbations, I would think from our experience, the variant function studies that we have been working on and more systematically also is really a reflection of what the community sees, which is that response QTLs are important to studies. Response means in the context of drug perturbations, stimulatory conditions, etc. So we observe when exposing cells to certain stimulatory condition during the differentiation process, and then performing stratified LD score regression, for example, and partitioning the heritability for insulin resistance or type 2 diabetes, that there's really an enrichment when cells are exposed to stimulatory conditions. So I would say that, it is really important to study natural genetic variation in the context of diverse stimulatory conditions in this sense. For the CRISPR perturbation, that's also a very good question, is I do think what would make sense, and the way we approach this is that we are working with a biobank of over 500 donors that hopefully increases over the next year substantially even further. And we can, we have aggregated, for example, genome-wide polygenic risk scores, aggregating millions of genetic variant into scores, and can identify individuals that are sitting in the middle of the score distribution or at the tail ends, etc., and can develop constitutively expressing DOX-inducible DCAS9 CREP or Cas9 lines out of those. So we have a good understanding of what is the genetic background that we are pulling or we are using for high throughput CRISPR perturbation screens, which I really think is really important. And one other note here is I think that the way we are developing the lines is also really important. When we think about monoclonal or polyclonal type of expansions, we can think that a monoclonal expansion is basically a result of a single cell state that we are that we are culturing in the dish. Which can be a large confounder. So I think attention is really important to these questions and understanding of what we are using for our CRISPR model systems.
Anyone else wants to add to this?
Well, I totally agree. Yeah, I totally agree with what Melina is saying, and we do not work with such well and deeply characterized biobank of donors, but we believe that everybody can have like this set of minimum characteristics. They're going to replicate the what the minimum necessary for the study of the mechanism. So yeah.
All right, I'll move on to the next question from Michael Nagel. Apologies if I'm pronouncing your surname wrongly. I tackled a Finnish name, but I struggle with English. Michael is asking, what are the panelists' thoughts about more complex cultures or 3D systems to understand function? Many genetic variants linked to disease play a role in inflammation, which many times require having resident tissue immune cells present in system to understand downstream consequences on target tissue. But I know this sounds very specific to immunity inflammation, but maybe from your experiences going beyond inflammation, how important it is to use more complex 3D cell cultures. Maybe I'll ask, Patricia, go ahead.
We like to look a lot into that. And as I mentioned, when David was talking about his imaging systems, which I think are perfect for that, right? When you're looking at in the tissue, for us, when we're working with immune responses, the interaction of cells is a major point. We are just starting to work with imaging of tissues for that matter. So we are doing that in fixed tissues and then trying to replicate that using a 3D cell culture where you can put together cancer cells and immune cells to understand the patterns of interaction and responses. And this varies a lot when you're working with 2D cell cultures and also with systems where you're doing co-cultures, different chambers. So I believe that the choice of the model is going to have a very big impact on what you're getting in the end, right? And comparisons of these different models is really important. So there is a lot of work being done nowadays trying to compare the different models for cell culture, specifically when you need to see interactions.
So kind of in a similar flavor, maybe I'll ask this question, I'll address this question to Melina. This is from Mike Payson. What are your thoughts on the trade-offs between systems with a single cell type versus allowing multiple cell types to interact? And you have previously mentioned the fact that you can't capture one of the cell states, and that's because of the interactions. So I wonder if Melina, you could extrapolate and expand on that a little bit more.
Yeah. So for our AMSC model, we understand now through these type of thorough characterization efforts that we are capturing different subpopulations of adipocytes, right, that are basically living together in a single well in our differentiation system. Additional to that, what Mike probably also means are co-culture conditions with, adipocyte, basically subpopulation of adipocytes and additional cell types. For example, immune cells, we know subgrade inflammation is a really important process in adipose tissue that kind of is important for disease pathogenesis. And I think co-culture systems are really, really valuable and invest efforts in, though I will say, I think these efforts are needed, understanding first through really deep molecular and cellular profiling, the individual cellular response to stimulatory conditions and differentiation dynamics is, I think, important to have confidence in a co-culture system where, the controlling of factors, where does a certain response in a given readout come from? Is it from the one cell or the other? Is not trivial, I think. So I do think, we have to be aware that in monocultures where we are looking at a single cell type, though reflecting different subpopulations, that there are limitations, of course, because cell-cell interactions with different cellular systems are so important to capture physiology and disease. And I would think that there's lots of effort needed to really build not only, culture cells together, but really have confidence in the outcomes.
And if I could maybe, David, ask your opinion on this when thinking about, other maybe non-transcriptional ways of phenotyping cells. Do you have experience with comparing monocultures to more complex models? How much are we recapitulating the biology or how much we're missing? How important are those more complex cellular models?
Yeah, I would say in in general, the need to have interactions between different cell types is important for understanding how these cells might actually function in the native tissue. Part of my lab studies macrophages, and most of the studies we do, we take a bunch of macrophages, differentiate them, throw them in a dish, and that's just very different than the macrophages that are actually there in tissues that are prepared to like respond to infections. The ones we use typically come from like a different developmental lineage, and then we also know that macrophages receive signals from like their neighboring cell type, from their neighboring cells that inform their function. So I think it is important. It's tissue-based studies are sort of where you actually have a chance to like study these things in their native context, but they also have their own limitations where you don't have access to perturbations, like you have with like the cell culture models. Mm-hmm. As far as like the co-culture models and combining them with profiling like lab cell imaging, I think like that can be a very powerful approach. The microscopes now are able to generate really nice three-dimensional data. I'd say being able to go from here's like my nice three-dimensional data set to here are nice single cell records of what each cell was doing over time, when they divided, who they were interacting with, when they were interacting, how their morphology changed. That step, I think, is something that's still right now like an open problem. And I'd say the big part is, we know that the algorithms like can do it, but the algorithms that, sort of are being commonly used for this require labeled data to learn how to perform these tasks. And the labeled data that's needed, right now just isn't there.
We have another question, which I'm actually not sure who to address it to. Maybe, David, you've got some experience with this. The question is, how important is measuring multiple protein expressions at minute levels, so nanoscales like femtomolars, versus what we currently have existing methods for like Olink array? Would that give a better handle to characterizing cellular heterogeneity? Anyone wants to take that question on?
Personally, I feel it's important. I would say like as far as its global importance, just based on the measurement technologies that my lab works with, the multiplexed immunofluorescence, I'd say it's a little unclear, just because most of the studies that people are doing are on like proteins that are fairly abundant, and to be able to measure the ones that are not abundant, typically you need to do some sort of amplification. And so the methods where you use like a bunch of amplification right now, those aren't commonly being used in the multiplexed spatial proteomics assays. So I'd say at least as far as my knowledge right now, sounds important, but I'm not sure.
Gosia, maybe I would like to add something to what David just said, because yes, of course, we usually work mostly with those proteins, but in our the systems that we work with, including response to infections, some proteins are expressed at very low levels at different times. So Olink, for example, is an interesting approach so that we are able to capture these small variations in time. So yeah, I think they are important depending on the system, just like David was pointing out, and we have some experience with that, the importance of measuring these very low levels of proteins within a timeline.
Great, thanks. So, we do focus a lot about sort of the challenges and the gaps, but I wonder if we could flip it around a little bit for the last maybe 10 minutes or so. And I wonder if you've got any examples you could share of actually things that have worked, of successes, and maybe in your opinion, where could we really now bring the community together and share the expertise, best practices, what in your hands you think we should already leverage and push forward in the community? Anyone would like to start? Helena.
Sure. I think my feeling there is that the successes, of which there are many in the IPS world, and despite all the challenges, they are and they remain a very powerful model to the field. But the success stories so far, to me at least, it seems that they have been achieved in the context of cell types for which we have a priori better understanding of the physiological relevance. So for instance, dopaminergic neurons, or in the diabetes field, kind of islet biology and so on. So I think the phenotype matters, and I think, Melina, you were talking about this either today or earlier, that kind of that in addition to focusing on things like transcriptomics and molecular measurements and even imaging measurements, I think we have to kind of think very hard what the physiologically relevant phenotypes are, and what we want to see, and can we see them in these models, and perhaps then that helps us to design better experiments, kind of aiming to capture that particular phenotype, as opposed to just differentiating cells towards a certain expected cell type, and then sort of, I guess, hoping that they will recapitulate what we would like them to.
Yeah, maybe I can add to that real quick. So I could not agree more. And I do think that, the question of which phenotypic readout to use is an important one. And I feel like beside molecular profiling, transcriptomic profiling, epigenomic profiling that we do regularly in the lab, what has been really helpful is to think about more downstream pathways and processes that we can capture and that are related to disease pathogenesis. And in adipocytes, insulin-stimulated glucose uptake is a measure of insulin responsiveness of these cells, and really helpful. And also just think about, this flow of information in a cell where we are starting from DNA and RNA and protein, and then the presumable convergence on a given cellular readout down the line, which in terms of success stories has been for us really helpful, because it decreases the noise in our systems when really, capturing cellular functions that are further downstream and, tightly linked to given disease relevant processes. So I think, I agree very strongly. And the other, in terms of a success story that I think has been really helpful, is our work with Cecilia Lindgren, where we are looking at histology-based measurements, and realize that the size of biobanks is really important, the number of donors to study natural genetic variation, but looking at phenotypes that are discrete phenotypes for measuring processes in a tissue, as for example adipocyte size estimates, appear to be really helpful as well, additionally to the in vitro systems that I mentioned before.
Any success stories from Patricia?
I am not sure which success stories, but we're working a lot with immune response related to cancer immunotherapy. And I believe there are very nowadays solid models for that based on the specific characteristics of the cells in the system. And this has helped a lot the community and treatment of patients. And this is based originally from what Melina and Helena were saying, of specific phenotypes of these cells that were originally used to characterize the cell lines of interest and the responses of interest. So I really believe that having this specific phenotypes of interest, which reflect the physiology of the cell and what you expect at different situations, is really important. And then from that, then you can move backwards to understanding which DNA and transcriptomics and regulation systems are in place, so that maybe, and in our case, is necessary to stratify later on cases in which there is no response, right? Because sometimes the phenotype reflects different levels of regulation within the cell, but we do have to first have that in mind, very well designed, and then move backwards to understanding. And that's moving really fast in cancer immunology. And I really believe this is a success story that we, for example, are working with, studying tumor microenvironment based on phenotypes and moving backwards to the mechanisms behind it.
Thank you. And then David, from your perspective.
I'd say from the imaging side, I think a big need has just been labeled data to power algorithms. And recently in our lab, we've been able to figure out how to label data with single cell resolution in a scalable way, so we can actually produce the data sets required to get these algorithms to work. I'd say, like, thinking back more broadly, I wouldn't necessarily call this like a full success in the sense that it's like completely done. But I would say, like, there are increasing efforts to sort of get the imaging data and then the non-imaging data together in the same place, and then do analyses where you can actually look, okay, what's the effectively like the mutual information shared across the different modalities. I'd say the best example I've seen of this to date has been work out of like Ann Carpenter's group, looking at like how much mutual information is there between cell painting assays and transcriptomic assays. I think she's speaking later today. But I'd say, like, I personally like would view that as being sort of the future of how imaging is going to be used to uncouple or to decipher like how diseases manifest. It's by integrating it with these other sources of data rather than keeping it sort of siloed.
Could I ask you to expand a bit more about when you say label data, because we spoke earlier about
Yes.
benchmarking how to, annotate your cellular models. And I can see how that would be really important for any machine learning and so on. So if you could expand more on that.
Yes. So the modern machine learning algorithms, differ from classical computer vision algorithms in like the following sense. The classical algorithms, the way in which they're developed is you get like a bunch of, very smart computer scientists. You give them a problem, tell them you want an algorithm that can do like A, B, or C. They explore the data, and then they effectively come up with like algorithmic rubrics for how to, get A, B, and C like out of the data. And something that the human mind came up with. In the more modern algorithms, the algorithms themselves like learn from data. So let's say a simple task like identifying cells in an image, rather than having like a human try to sort of verbalize in code speak what a cell looks like, rather instead you would take a bunch of images, circle a bunch of cells, hand them to the algorithm and say, hey, this is labeled data about what cells look like. Can you learn on your own how to do this task? And so we just know from experience now that these new machine learning approaches can be very powerful and very accurate, to the point where you can almost automate particular types of analyses. The downside is that they do need this labeled data in order to work well. And I see, just broadly speaking, the more labeled data you have, the better. The more diverse labeled data you have, the better. And it's essential to get these to work, but it is, I'd say, like quite painstaking to generate. So yeah, I think that at least as far as the intersection of machine learning and biological imaging, I'd say like that's been like a big barrier, a big bottleneck, and a big challenge. It still is a big barrier, a big bottleneck, a big challenge. But I think we're starting to see, some of the early data sets like come online, that are going to let you pull the sorts of information that you want out of images. Example, like we work with some folks at Stanford, Mike Angelo, on doing single cell analysis of tissue imaging, being able to interpret tissue images has been with single cell resolution, both identifying cell nuclei, but then also identifying like the whole contours of each cell. That's something that's been like a major challenge. Working with Mike's lab, we were able to generate a data set where we labeled over 1 million cells, both their whole cells and also nuclei, then use it to generate machine learning methods that can do the cell identification task with effectively human level accuracy. And right now, like my lab is working on porting over some of these methods for a variety of different challenges for live cell imaging data. But it's it's exciting times, but there's a lot of work to do.
Great, thanks for that. And we have just a couple of minutes left. There's one question that actually lands close to my heart, and that's Myla Prokunina Olsen is asking about how do we address transient and spatial effects. And we in my lab, we work on immune responses, and something that we recognize particularly important and relevant is mapping those cell state dependent EQTLs. And we see that when we're missing the colocalization between GWAS variants and EQTLs, having those detailed maps of how gene expression regulation happens as cells transition through different states, for example, T cells in response to activation, is really informative and allows us to map those effects that we don't see in steady state. So how do we address that? From my perspective, we need to start really thinking more about going beyond the steady state measurements and start thinking more about measuring phenotyping cells in response to different stimuli throughout different time points of development and so on. This has been, I'm just looking at the time, so sorry, I've stolen the last question. If panelists wants to pick up on this, please go to Slack and comment on this. This is a great question. Thank you very much for this discussion. I think there's been a ton of really informative discussion here. Quite a few points that are emerging and themes that I hope we can take on as a part of ICDA, as an action point, as a recommendation, and help the community to move forward. So thank you all once again. We are taking a break